Hello, and thank you for virtually attending the 2021 Alzheimer's Disease Genetics Global Symposium, Pathway to Translation. I am Badri Vardarajan, Assistant Professor of Neurological Sciences at Columbia University, and I will summarize the salient points of discussion from the symposium. The symposium comprised of 5 sessions: genetics, discovery and risk scores, genes and environment across populations, biomarkers and endophenotypes, towards precision medicine and therapeutic targets. The first 2 sessions discussed genetic findings in Alzheimer's disease, cognitive decline, endophenotypes, and discussed the modifiable environmental risk factors in diverse populations. Several epidemiological and genetic studies in minority and non-European populations were also described. The biomarkers and endophenotypes session detailed the recent advances in fluid and imaging-based biomarkers for early and accurate detection of AD and related neurodegenerative diseases. Multi-omic markers, including the metabolome and the microbiome, were also discussed in detail. The precision medicine session comprised of a discussion about multi-omics, translational and model systems research, and clinical and genetic counseling to realize the bench-to-bedside goals of the precision medicine in AD. The last session on therapeutic targets summarized the research in several independent and druggable targets identified by genetics and transcriptomics and provided a comprehensive view on the current therapeutic landscape of Alzheimer's disease. I'll summarize the key takeaway points from this symposium. The first being the role of diverse cohorts in understanding the biology and treatment of Alzheimer's disease. The first 2 sessions, as I mentioned earlier, focused on how large-scale genome-wide association studies have and are still continuing to provide insights into the genetic risk of Alzheimer's disease, highlighting previously little-known pathways such as innate and adaptive immunity in the pathogenesis of the disease. A couple of talks also focused on the utility of polygenic risk scores, particularly in minority populations. Polygenic risk scores could identify at-risk individuals and facilitate early interventions, a stated goal of precision medicine. However, a major limitation of the genetics field in general is the Eurocentric bias in genome-wide association studies. As the figure shows, according to the GWAS catalog, about 80% of the GWAS participants are of European descent, despite making up only 16% of the global population. This is particularly problematic because previous studies have shown that African American and Hispanic individuals contribute an outsized number of associations relative to the studies of similar sizes in Europeans. It is encouraging, however, to note that within Alzheimer's disease, there is a concerted effort now to conduct well-powered large studies in Asian, African, and South American populations. And some of these efforts were extensively characterized and discussed in the first 2 sessions of the symposium. The second major area of recent development in Alzheimer's disease is the identification of fluid biomarkers in CSF and blood to facilitate early diagnoses, risk, and severity assessment of Alzheimer's disease. The figure illustrates neurons and AD pathology with the corresponding AD-related biomarkers in text. For instance, CSF and plasma A-beta 42 to A-beta 40 ratio, as well as amyloid PET imaging, are direct markers of AD pathology. The recent development of blood tests identifying the phosphorylated tau 181 and 217 as an early biomarker of AD pathology could prove as a valuable tool for early detection. It's less expensive than PET imaging and less invasive than spinal taps. And these blood-based biomarkers can distinguish Alzheimer's disease from controls and also from other non-AD forms of dementia. However, more studies in diverse populations and different stages of the disease are still needed to validate these findings. But these blood-based biomarkers have the potential to become the gold standard test of detecting AD early and efficiently. More work into identifying biomarkers that can detect AD and concomitant pathology early in the disease course is thus strongly warranted. Another significant area of discussion was to leverage the findings from genetics and multiomics to identify druggable targets in Alzheimer's disease. Amyloid and tau aggregation and cholinesterase enzyme pathways have been the go-to areas for drug target development. However, a recent spate of genetic findings pointing towards innate immunity, inflammation, and oxidative stress pathways have opened up several potential areas of target identification. The therapeutics session discussed several such approaches, moving above and beyond the amyloid cascade pathways. The figure shows compounds in clinical trials in 2019 for treatment of Alzheimer's disease, characterized by their desired targets. Several phase 1 and phase 2 clinical trials are now target pathways beyond the usual suspects, amyloid and tau. The identification of several interconnected pathways in AD is thus leading the research towards multi-target drugs with the aim to target several processes that contribute to Alzheimer's disease simultaneously and thus limit their role in the development of the disease. I hope that the topics covered in the symposium were informative to the researchers, clinicians, and the caregivers, and it could spur more collaborative research to understand and alleviate this debilitating disease. On behalf of the organizing committee, I thank the speakers, the participants, the National Institute of Aging and Alzheimer's Association for making this exciting symposium possible. We look forward to your participation in the Q&A session, and hopefully we'll see you again next year.